Just waiting for a confirmation that the live stream is there. If you can see me live, just put in the comment below. If you can see the live stream. Okay, now I can see myself live, right? So let's start this session. So again, like all YouTube classes, first what I will do is I'll show you a few questions on hexomenophosphate shunt, so that you have an idea as to what kind of questions are asked. Then we will look at the theory portion of it in a revision mode. So only the most important point, most important concepts, and then again we will come back to the questions. Okay, so uh, in the live chat you have to put all the question, all the answers of the quiz. So let's look at the first question. Which of the following is false about HMP shunt? Try and answer. So all of you know that there is a live chat option. Just keep putting your comment below. Which of the following is false about HMP shunt? No ATP formation, formation of NADPH, synthesis of ribose 5-phosphate, none of the above. Okay, so Gopal says D. Good. We will come to these questions, don't worry. Let's look at the next question. Which of the following cellular organelle does HMP shunt occurs? Very important question. Where does HMP shunt occurs? Does it occur in nucleus, in mitochondria, in lysosome, or in cytosol? Where does it occur? Again, a very important question. So Manisha, Satish. Hi Manisha. The storage disorders, there will be a session in this month. Both are Unacademy 1 hour session and a half an hour YouTube sessions. Both because I remember you have requested for storage disorders. After that you will remember all the storage disorders. Okay, so just be tuned in. Okay, all the storage disorder, lysosomal storage disorders, all the what you call mucopolysaccharidosis, glycogen storage disorders, all I'm going to cover. Okay, good. And similarly I have taken all the whatever students have requested in this month. Sites at which HMP shunt can occur include all of the following except. Sites at which HMP shunt can occur include all of the following except. So options are liver, adipose tissue, adrenal cortex, kidney. I'm not giving the answers because this is just warm-up questions so that you can see how much you know about these topics. Okay, Gopal says D. Manisha says okay. Right. Let's look at this question. Main source of NADPH is, again, main source of NADPH is it glycolysis? Is it TCA cycle? Is it pentose phosphate pathway? Is it gluconeogenesis? So NADPH, where it is produced. So Arjun says D, gluconeogenesis. Gopal says C. Satish also says C. Okay. Now this is a very, this was asked in names. Which of the following step of HMP pathway requires thymine pyrophosphate as cofactor? So thymine pyrophosphate as a cofactor is utilized in which step? Is it glucose 6-phosphate dehydrogenase? Is it 6-phosphogluconate dehydrogenase? Is it trans-ketolase or is it trans-aldolase? So Manisha says trans-ketolase. Anyone else? Gopal also says A. Arjun also says C. Okay. Guys, all of you please answer. Don't worry even if you get it wrong, right? Because there is something called as participatory learning. You learn more when you participate. So there are so many students who are live but they are not replying. Your learning will be much lower when you reply. Okay. And trust me, now in a live class it is difficult to participate because that peer pressure is there. You have to be right always, or are sometimes we feel that we should not look fool in front of other friends. But this live platform, you should participately, in a very aggressive way. Okay, all are true about hexose monophosphate shunt except, so you have to find the false statement. Produce NADPH in oxidative phase of pathway, does not produce ATP, produces ribose 5 phosphate in oxidative phase of pathway, or glucose 6 phosphate dehydrogenase enzyme is involved. So which of the following is a false statement? Okay, so metabolites in HMP shunt are all except. So which of the following is not a metabolite in HMP shunt? Is it glycerol 3 phosphate, acetoheptulose 7 phosphate, glyceraldehyde 3 phosphate, or xylulose 5 phosphate? Good, Manisha. So everyone is saying glyceraldehyde 3 phosphate. Gopal is saying glycerol 3 phosphate. Anyone else? Satish is also saying glyceraldehyde 3 phosphate. Anyways, so now we will look at the theory. So first 5 minutes I generally take to just warm up students so that they. So let's look at the theory portion of HMP shunt. Very important, very small but very important topic. So it is also called as pentose phosphate pathway. Okay, because 5 carbon sugars phosphates are being formed. Also called as Dickon-Horowicker pathway or phosphogluconate pathway. So all these three are alternative names of HMP shunt. Very important, very frequently asked. It happens in cytosol, and the rate limiting enzyme is glucose 6 phosphate dehydrogenase. Now this is an alternative route for oxidation of glucose. We know that glucose is converted into glucose 6 phosphate, okay, and this glucose 6 phosphate can take a lot of route. It can go to glycolysis, okay, it can, which is an oxidative pathway. It can go to glycogen synthesis, okay, glycogen synthesis, or it can even go to one of these pathways, which is HMP pathway. Okay, so it has a lot of alternative route way. So it is one of the alternative route of oxidation of glucose beside glycolysis. But the importance here is it does not generate ATP. So it's not an ATP generation pathway. Now you have to understand this simple concept. Glucose, when it comes, it's an energy rich molecule for cell. Now cell breaks some part of it for immediate energy release, and whatever additional it has got, it can store as glycogen, it can convert into fatty acid stored there, okay, or it can produce reducing equivalent like NADPH, okay. So it is also a form of energy. Remember, this NADPH is also a form of energy. So it does not require immediate energy because ATP is for immediate energy. So what it can do is it can change this glucose 6 phosphate in NADPH form, which can go to electron transport chain, and there they can generate energy also. And also it is very important for providing 5 carbon sugars. Now these 5 carbon sugars are very important for nucleic acid synthesis, both RNA and DNA. Okay, so this is the main function of what you called HMP shunt. So it provides 5 carbon sugars and it provides a lot of reducing equivalent, which is very high energy. Okay, each NADPH, if you remember from our electron transport chain lecture, each NADPH will produce equivalent to 2.5 ATP. So this is a very rich way, very compact way in which energies are stored. Okay, now what is the split between glycolysis and HMP shunt? 90% of the glucose will be utilized by glycolysis and around 10% only goes for HMP shunt. Okay, however, in liver and RBC there is a difference. Okay, 30% of the oxidation will happen, will go to HMP shunt in case of liver and RBC. So liver and RBC are very hugely. Can anyone tell me, liver we understand, can anyone tell me why RBC lot of HMP shunt is happening? In RBC, why are we having a lot of HMP shunt taking? Almost 30% of the glucose is going for HMP shunt in RBC. Anyone? Can anyone give me the reason for this? These NADH are important, very good, very good, very important to keep the iron in the reduced state. And if this is not there, then the RBCs will go into hemolysis. So very good, very good. Glutathione, all of you are correct in glutathione. Sesha, Manisha, all of you are correct. Okay, so this is why liver will have and RBC will have almost 30% of the glucose going to HMP shunt. Okay, now let us look at what is the absolute basic of HMP shunt. So what happens, glucose 6 phosphate with 6 NADP will form 2 glucose 6 phosphate, 1 glyceraldehyde 3 phosphate, and along with 6 NADP and 6 carbon dioxide. So this is the overall reaction which is happening. Okay, this is the overall reaction of glucose of our HMP shunt. Now obviously there are so many intermediates, and the main function of HMP shunt is to produce a lot of intermediates which can go into the pathway. But overall, if you see, the generalized reaction is glucose 6 phosphate, okay, is converted into 1 glyceraldehyde 3 phosphate, 3 carbon dioxide, so carbon dioxide is produced, okay, and 6 NADPH. Okay, now here we can easily compare between glycolysis and HMP shunt. As you can see, in in glycolysis, ATP is formed. Here ATP is not formed. In HMP shunt, carbon dioxide is formed. Here it is not formed. And here 5 carbon sugars are there. Here it is not there. So these are the major differences. There are many other differences, but these are the major differences which you have to be there. Okay. So let us look at the phases of HMP shunt. First is the oxidative, but it is also the irreversible phase. This happens in liver, adipose tissue, adrenal cortex, erythrocytes, gonads, and thyroid, and only in lactating membrane. It's irreversible, and this is the place when NADPH is required. Okay, so dehydrogenation of glucose 6 phosphate will lead to decarboxylation and production of ribose 5 phosphate. Okay, now remember, dehydrogenation, a very important concept is that NADP is used, not NAD is used as an acceptor. Many a times they will give NAD and students confuse. It is NADP which is used as a hydrogen acceptor, not NAD. Okay, so what happens, glucose 6 phosphate with NADP will form, and 6 phosphogluconate, which will be converted as with the help of the same enzyme decarboxylase into ribose 5 phosphate, and here 1 carbon dioxide will be coming out. Okay, so this enzyme, remember, glucose 6 phosphate dehydrogenase is the rate limiting enzyme. Okay, and this is a irreversible phase. This is a irreversible, oxidative irreversible phase. Okay, so the second is a non-oxidative phase. This happens in rapidly dividing cells. Okay, so as we have seen that a reversible oxidative phase, the main function was to produce 5 carbon sugars and NADP. Here what will happen, we will have a lot of intermediate 5 carbon sugars. Okay, so that's why it is there in rapidly dividing cells like bone marrow, skin, mucosa. This is a reversible phase. Remember, this is a reversible phase, and what happens, it has two kind of reaction. One is a trans-ketolase reaction. So in trans-ketolase reaction, what happens, it requires thymine pyrophosphate and 2 carbon fragment are moved. I'll show you the reactions. So what happens, 2 carbon moiety is basically moved from one to other. Okay, aldolase, it will transfer 3 carbon moiety and it does not require thymine pyrophosphate. So once we are going to see the reaction, there we can see. Okay, so trans-ketolase, trans-aldolase. Now let's look at the non-oxidative. So what happens, ribulose 5 phosphate with the help of isomerase, okay, will be forming either ribose 5 phosphate or xylulose 5 phosphate. So this ribulose 5 phosphate is forming ribose and xylulose. Okay, so it is in the non-oxidative phase, that ribose 5 phosphate is formed. Now the first reaction happens, which is a trans-ketolase reaction. Trans-ketolase, as I told you, what will happen in this trans-ketolase reaction, this is a 5 carbon. Okay, this is 5 carbon. Now trans-ketolase moves 2 carbon atoms. So what it will do is, it will move 2 carbon from here and give it to xylulose. So this 5 carbon will be converted into a 3 carbon glyceraldehyde 3 phosphate, and this 5 carbon xylulose will be converted into a 7 carbon pseudohoptulose. Okay, so this is what is happening, trans-ketolase. Very important, thymine pyrophosphate is a coenzyme for trans-ketolase reaction, number 1. And this is the step which is inhibited in Wernicke-Korsakoff psychosis. We are going to study in the clinical correlation. Then the next step is when there is a trans-aldolase reaction. So what does trans-aldolase do is, as I told you, this does not require thymine pyrophosphate, and there is a transfer of 3 carbon. So now this 7 carbon, what it will do is, and this is 3 carbon, so it will transfer 3 carbon back here. Okay, so now 3 plus 3, it will become 6 carbon, fructose 6 phosphate, and this 7 carbon will be only 4 will be left, so it is erythrose 4 phosphate. So again, now this is a 4 carbon moiety. And again, in the last step, what will happen? Again, trans-ketolase reaction will be happening. Okay, and this will be converting into 6-phosphogluconate, and yeah. So this is what is going to happen. This is a non-oxidative. Now you don't require to remember all these steps. There are few things which you have to remember. One, and I am going to just enumerate those things. One is trans-ketolase moves 2 carbon, trans-aldolase moves 3 carbon. Number 1. Number 2, trans-ketolase requires thymine pyrophosphate, trans-aldolase does not require thymine pyrophosphate. And number 3, the most important is, what are the intermediates in non-oxidative phase? Okay, so ribose, xylulose, glyceraldehyde, cetoheptulose, erythrose. Okay, so these are the intermediates of non-oxidative phase. Okay, very good. Okay, so this we have covered. Now let us look at the biochemical significance. So obviously we have seen it's an alternative route of metabolism of glucose. There is a complete oxidation of glucose, and this is a very complex pathway, much more complex than glycolysis. The major function is it generates NADPH and ribose. There is no ATP generated, and the pentose derivatives are useful for synthesis of nucleic acid. Also, this NADP which is produced is required for reductive biosynthesis of fatty acids and steroids. Okay, so and microsomal P450 liver also utilizes this NADP to become detoxification of drug. So let's look at 2 clinical correlations. First of all, glucose 6-phosphate dehydrogenase deficiency. You are going to read a lot about this in PATH. It's an inheritance X-linked recessive disorder. Okay, now the basic pathophysiology, what happens in HMP is the only means of providing NADP in erythrocyte. So decreased activity will impair synthesis of NADP, which will lead to accumulation of methemoglobin and hemolysis. Okay, and this manifests as hemolytic anemia and methemoglobin. So already we have talked about it, and there are certain drugs which can further reduce the activity, and that will lead to other further hemolysis. The second clinical correlation, which nobody which has been asked quite a few times, is Wernicke-Korsakoff syndrome. Here there is an alternation of trans-ketolase activity which reduces the affinity with the thymine pyrophosphate. Now remember, another very important question is, if we want to see the activity or level of thymine pyrophosphate, okay, we measure the trans-ketolase activity in the RBC. So this was again a AIMS question. So how do we measure the level of thymine pyrophosphate activity? It is by the trans-ketolase activity in the RBC. And again, Wernicke-Korsakoff syndrome, because this trans-ketolase activity is not happening, there will be mental disorders, loss of memory, and partial paralysis. So these are the 2 clinical correlation. So I've already told you these clinical points. Now let's again get back to the question. So last 10 minutes, let's quickly solve the questions. Which of the following represent the primary function of pentose phosphate pathway? So what is the primary function? In erythrocyte. So which is the primary function of pentose phosphate pathway in erythrocyte? Quickly, can anyone answer? So you have to be very fast. Very good. All of you are correct. The correct answer is production of NADPH. Okay, the production of NADPH. So this is the correct answer. Very good. Let's look at this. The reducing agent used in lipogenesis is derived. Now these are all actual exam questions. Is derived from pentose phosphate pathway, glycolysis, TCA cycle, gluconeogenesis. Very good. So I'll not spend a lot of time. The correct answer is pentose phosphate pathway. My idea is that you should get a coverage of all the previous year exam questions. Ribose phosphate pathway is a product of which of the following pathway? Again, I'll skip this question. I'll not put a lot of here. It is pentose phosphate pathway. Now this was again an AIMS question. Thymine level is best monitored by. So this was the question which I was talking about. It has come in AIMS. Thymine level is best monitored by trans-ketolase activity in blood, thymine level in blood, G6PD activity, or reticulocytes. So what should be the answer to this? Very good, very good. The best, one of the best monitoring method. So this was the question. Many students actually went for thymine. Many students, went for thymine level. The correct answer is transketolase level in blood. Very good, Lokesh. Very good, Arjun. Very good, Manisha. Okay, the rate limiting step in HMP shunt is. Quickly answer this. Which is the rate limiting step? So is it glucose 6 phosphate to 6 phosphogluconate, 6 phosphogluconolactone to 6 phosphogluconate? Okay. So correct answer, very simple, it's A. Okay. Glucose 6 phosphate dehydrogenase needs. Now this was the question I was talking about. What is the cofactor or which is the acceptor of hydrogenase? Quickly answer this question. Glucose 6 phosphate dehydrogenase need. Rifaka, this is wrong. Remember, anyone else wants to answer? Arjun, it is correct. Very good, very good. Remember, very important MCQ. Very important MCQ. It is NADP, not NAD. Don't get confused. It is NAD. Okay. Very good, very good. Okay. In which condition does hemolysis occurs on oxidation? Again, it's a very simple question. I'll not spend a lot of time. It is G6PD deficiency. Very good. Yes, this is a good question. Try and answer this question. In non-oxidative phase, ribulose 5-phosphate is isomerized to. Quickly answer this question. In non-oxidative phase, ribulose 5-phosphate is then isomerized to. So what is the isomerization? Very good. The correct answer is ribose 5-phosphate. Okay. So the second transketolation reaction, involves. Try and answer this question. This is a very tricky question. So it clearly says, I have told you that true transketolation reaction. First, so what happens is there is a transketolase reaction followed by transaldolase, and last again it is transketolase. Okay. So what is this transketolase reaction? So B is wrong, Satish. So remember, so transketolase, so first of all, in transketolase, 2 carbon will be moved. Okay. So first let's, even if I don't remember it, what is happening? 4 is becoming 5, and so here 1 carbon movement is there. So this cannot be a transketolase. Let's look at this. 3 is becoming 4 and 7 is becoming 6, right? So again, how many carbons are being moved? 3 carbon is being moved. 3 carbon is moving. 3 is becoming 6 and 7 is becoming 8. So again, this cannot be a transketolase reaction. Let's look at this. So 5 is becoming 3 and 4 is becoming 6. So 2 carbon is being moved. So this can be a transketolase, and here again 5 is becoming A. So both of these can be transketolase ideologically, but we know that 5 carbon is not, it is 5 4 6 3. So the correct answer should be C. Even if I don't know it, I could have got it correct just by knowing the fact that in ketolase reaction 2 carbon is moved, and in aldolase, okay, in aldolase 3 carbon is being moved. Okay. Now clinical aspects of HMP shunt include. Quickly, a very simple question. So all the 3 will be many people just mark G6PD deficiency. The correct answer should be D. Okay. So I will not. Okay. This is a very good question. Maximum concentration of NADPH is seen in. Maximum concentration of NADPH is seen in. Try and answer this question. It's a little different question. So which of these organs need absolutely zero, has zero tolerance for NA? Very good, Satish. Very good, Satish. You have got it correct. lens of the eye has to be absolutely transparent, and that entire thing is focused on. So the correct answer is lens of the eye. Very good. Maximum concentration of NADP. Okay. So genetic defect of enzyme transketolase, again very simple, Wernicke-Korsakoff syndrome. Okay, let's look at some of the reason assertion question. Assertion is about 10% of the glucose molecule per day are entering into HMP pathway. This pathway means HMP pathway. Okay, HMP pathway. The liver and the RBC metabolizes 30% of the glucose by HMP pathway. So what should be? Should it be A, B, C, D? Very good. So both these statements are actually correct, but this is not the correct explanation. This is not the correct explanation. So answer should be B. Very good, Manisha. Very good, Arjun. Let's look at this question. Transketolase transfers 2 carbon unit from xylulose 5-phosphate to ribose 5-phosphate to form a 7 carbon pseudoheptulose and glyceraldehyde phosphate. So what is the transketolase enzyme will transfer a group of from a donor ketose to an aldose acceptor. Is it true or false? So what should be? So this is absolutely correct, Gopal. So both these statements are correct, and actually this is the correct. That is why it is transferring 2 carbon. Okay. So this is, both of them are correct. Very good. So the correct answer is A. Okay. Now this question, again very important. This pathway means HMP pathway. Okay. HMP pathway is mainly regulated by level of NADP. Okay. And 6-phosphogluconate dehydrogenase is the rate limiting enzyme. So what is correct? Quickly, anyone? So what is the rate limiting enzyme? Just put it in the comment. Good, all of you are correct. The correct answer is, this is correct, but this is false. So it will be A. Let's do this match the following, because match the following is very important for AIMS. So A, quickly, pentose phosphate pathway. So whichever you are sure, A1, you can just put it. Very good, Manisha. A4, so pentose phosphate pathway. No, look, you have to be very, very sure, because something, they can be more than one A. Dinesh, you can always see the recorded version of this session, and you will be able to answer most of this question. So please just put what we are saying. So B5, very good. Look, I think okay. What do we have? So transparency of A. How about non-oxidative phase? One, we are very sure transketolase is a thymine diphosphate. This is very, very clear. Okay. C3, not very good. This also is clear. Okay. So C3. Okay. Transketolase D2. Very good. Maya, it will be D2 because there are multiple correct A. So that's why it is correct. Transparency of lens, I think it should be so what are we left here? Transaldolase. NADP and pentose phosphate pathway. So if I have to choose, probably I'll pentose phosphate pathway can actually go for ribose A. NADP can be for transparency of lens, and trans-aldolase is a Schiff base. Okay. So sometimes you have to start with one of the most obvious A, and then you have to, because sometimes one can match with more than one. Let's look at some of the true and false before we end it. Measurement of erythrocyte transketolase and its activation by thymine biophase produces an index of vitamin B nutritional status. So this is a true statement. So transketolase converts ribulose 5-phosphate to its converse aldopentose. Is it true or false? No, this is false. It does not convert into a pentose because this itself is a fire. Transcatolase will convert it into a cell. So this is false. No ATP is generated in pentose phosphate pathway. This is true. Trans-aldolase catalyzes transfer of 3-carbon dihydroxyacetone moiety. Very good, Saira. So this is true. Trans-aldolase. Pentose phosphate activity is high in non-lactating mammary gland and skeletal muscle. Is it true? This statement, please answer for this statement. This is a very important here. I want an answer for this because there is always a delay in YouTube transmission. Please answer for this. Very good, very good. All of you have got it. Rifaka, this is fault. It is non-lactating mammary gland. If it was lactating mammary gland or so in skeletal muscle, pentose phosphate pathway is one of the lowest. Okay, so this is a false. Remember, good game. The hydrolysis of 6-phosphoglucanate is accomplished by gluconolactone hydrolase. Is it true or false? The last question. Very good. All of you have absolutely, got all the questions correct. So this brings us to the end of our class. Now, what I will request all of you, this month's YouTube session I have already planned, but still, if there is any high topic which you want me to cover, you can directly reach me through Unacademy app. You can directly message me here. I read all the comments. So please put in your comments any topic which you feel. So because that helps me to plan what topic you are on. These are all free sessions. You can all take the benefit of it. And if you all feel need to join Unacademy, because live teaching is much better. You are here, you don't have to prepare anything like a class. I'll teach, I'll discuss questions, you can ask problem. So if any one of you are willing to join Unacademy paid programs, you can use this code Dr. Abhishek 10. Okay, so what happens is you will get a minimum of 10% discount. It can be even more if there is some promotion going on. And second thing is I'll get a credit for it. Okay, so let me be very, very upfront. Whenever you make a payment using this code, I get a certain portion of the year. So I will be very transparent, but that money is not very important. Okay, it's a very small fraction. The more important is we get an accreditation that so many students have liked our teaching and they have used our code. So with this, okay, protein techniques, very good. So this is a very important protein separation techniques. I think you mean by Saira. Protein separation techniques. Okay. Very good. So that I'll definitely plan. So guys, just put all your comments here, and I love to definitely you will see that I'll plan something either in form of a special class or a YouTube class. Okay. Right. Take care, guys. Thank you. You have been so lovely. Take care. Bye.